Get into our project, some housekeeping announcements. Restrooms are down the hallway next to the staircases on the right-hand side. Please do not eat or drink in the auditorium. And for our Zoom audience, this meeting will be recorded, so if you do not wish to be recorded, please log out.
We would like to now present the results of the research we have been conducting for all of the fall semester. Starting with our undergraduate trainings, we will start with Colby Macapago. Go Colby!
This one.
Yeah.
So this is Zoom audience. Can you please tell us whether you can see us and hear us?
Yes.
I see you and hear you, Dr. America.
Thank you.
Okay. Hello everybody. To reintroduce myself, my name is Kobe H. Makbagal, and today I'll be introducing to you my project on mapping the risk, identifying leptospirosis hotspots across the world. So to start off, it's important to know the main character of my project and what leptospirosis is. I do think its etymology shares enough information about it, where lepto means thin and spira means coils. And these thin coil bacteria spread through animal urine that contaminates the environment. Whereas seen in this video, it can enter exposed individuals by corkscrewing through cuts or mucous membranes if digested. Here at home, Hawaii has the highest infection rate in the United States, and people are exposed while hiking, swimming, dealing with animals, and farming. And you're probably familiar with the signs posted by streams and waterfalls and on the news. The aim of this project is to assess the presence of leptospira in water and soil in Oahu. Just a quick note, leptospira is the bacteria that causes leptospirosis. And to do this, my project has three objectives. First, collect samples from predetermined hotspots. Second, develop a standardized quantitative PCR, or a qPCR for short. And lastly, identify positives and map via ArcGIS software. What makes this project important is that currently leptospira prevalence is typically seen in only water samples, and that the data for specific variables and locations such as analyzing soil and Oahu's forest reserves are few, which makes it harder for clinicians, farmers, and residents to understand where risk is highest. This leads to more problems such as underdiagnosis or underreported infections, which at worst can cause organ failure and death. In this battle against leptospirosis, my project fills these gaps to better support public health awareness, prevention, and mitigation for communities that interact with these environments every day. So to do this, we collect water and soil samples from streams' upper, mid, and lower areas. When we were in the forest reserves, we had permits from the Department of Land and Natural Resources. With these samples, we then filtered to isolate leptospira, extracted the DNA for further isolation, and then PCR to amplify the LIP-L32 gene of interest. We then analyzed the sample's quantification of leptospira, then mapped the positives accordingly. For accurate detection and quantification, all the results are standardized using a qPCR standard curve. Instead of just reporting CT values, we convert every sample into genome equivalents, or GE for short. So for example, if a sample gives a CT of 18, that corresponds to about 260,000 genome equivalents per liter. The standard curve is essential because it also checks that our primers and reagents are working correctly. Each run should have the same slope. If the slope changes, something failed and the data isn't reliable. So in short, converting to GE gives us more accurate, repeatable, and meaningful quantification of leptospira in the environment. A Upon mapping, we found that the highest leptospiral loads occur downstream in urban and recreational areas. So an example would be the Alawai Dog Park, so down here. Soil is a persistent reservoir with several sites showing soil-only positives, even when water was negative. And looking at this table, we can see that 7 of the 17 water samples tested positive, and 8 of the 17 soil samples also tested positive. There was a prevalence in the Manoa Stream, Nu'uanu Stream, North Fork, Kauanoa Hua Stream, Monowili Stream, and Waimano Stream. All these areas right here, 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 and here. Also here. From this data, we also saw that the Alawai Manoa downstream had the highest water contamination, and the Kailua Beach downstream of Monowili Stream had the highest soil contamination of leptospira. In conclusion, this project revealed that downstream urban and recreational areas show the highest leptospira loads, indicating elevated exposure risk. Soil is a persistent reservoir with positive hits even when water is not, highlighting overlooked transmission potential, and that qPCR and GE results improve detection accuracy, allowing clearer identification of high-risk sites. Moving forward, our work includes expanding seasonal sampling, observing the effects of precipitation in leptospira transmission, incorporating wildlife surveys, and developing spatial risk maps to guide public health efforts. I'd like to thank my mentors and lab partners, Dr. Vivek Nurakar, Dr. Rodson Zarilla, and Lynn Tran. And I'd also like to thank the MHRT team with Dr. C and Dr. Toll, as well as funding from IMBRI, and the NIH and MHRT as well. Thank you. So our next person up tonight is Amy Dennis.